Hi, today we will be talking about transcription and translation in eukaryotes with Caroline and Elizabeth. First, let's look at the DNA that will be transcribed. It is double-stranded and anti-parallel, so the strands go opposite directions. In transcription, DNA is read 3' to 5', so we'll be transcribing the template strand. We begin transcription with initiation. In initiation, RNA polymerase and other transcription factors bind to their promoter region. In eukaryotes, this region is called the TATA box. RNA polymerase is an enzyme that creates the mRNA transcript from DNA. After initiation is elongation. This is when RNA polymerase starts synthesizing the mRNA, which gets synthesized in the 5' to 3' direction. In terms of bonds, there are phosphodiester bonds between the nucleotides on both DNA and RNA strands. Hydrogen bonds form between the base pairs, either DNA and DNA pairings or DNA and RNA pairings. Next is termination. This is when the terminator sequence tells RNA polymerase to stop and it releases the mRNA transcript and lets go of the DNA. The terminator sequence does not get transcribed. Before we can move on to translation, we must first do RNA processing. In this process, a 5' guanine cap is added to the 5' end of the mRNA, which helps the ribosome find the transcript and protects the 5' end when it moves out of the nucleus. On the 3' end, a poly-A tail is added, which helps stabilize the transcript and protects the other end. Splicing also occurs in RNA processing. This is when spliceosomes remove introns, which are non-protein coding sequences, and connect exons, which are protein coding sequences. Once RNA processing is complete, the mRNA transcript moves out of the nucleus and into the cytoplasm where translation will take place. In order for translation to occur, we need a ribosome. Ribosomes have large and small subunits as well as 3 different sites: the exit site, the peptidyl site, and the aminoacyl site. The first step in translation is initiation. This is where the ribosome locates the mRNA transcript and scans for the start codon, which is AUG. After this is elongation. As the ribosome moves to the next codon, the next tRNA with the matching anticodon carrying the next amino acid enters the A site of the ribosome. It then moves to the P site where the amino acid is added to the growing polypeptide. The tRNA, now with no amino acid, goes into the E site and exits the ribosome. In this process, hydrogen bonds are created between the codons on mRNA and anticodons on tRNA. Peptide bonds are made between the amino acids on the polypeptide. The last step of translation is termination. This occurs when the release factor recognizes the stop codon, which is UGA in this case, and causes the translational complex to dissociate and the completed polypeptide gets released. And that is all we have on transcription and translation. Thanks for watching!